Okay, so I think we'll go ahead and get started. Since our time is a little bit limited today, and we're trying to pack in a lot of information. So welcome everyone to the first of our series on respiratory pathogens. And today we're going to be discussing influenza and the influenza research database. So welcome, my name is Virion Structure, and I'm a bioinformatics analyst and the outreach coordinator here at JCVI for IRD and the virus pathogen resource. And with me today are Yun Zhang, a senior bioinformatics analyst, and Richard Sherman, the director of informatics at JCVI, who will be helping me moderate this series and answer any questions. We also have our guest lecturer, Dr. Daniel Perez, who will be talking to us about fun with influenza viruses. So just to give you an idea of the structure, I'm going to just give anyone who might not already be familiar with influenza viruses and Orthomyxoviridae a quick introduction to them. This will be followed by our guest lecturer's talk, leaving some time for questions, and then I'm going to move on to a live demo and tour of IRD and just highlight some of the most important features that we have, and we should end by 9:30. So just to give you a little bit of background on who we are, we were established, the bioinformatics resource centers were established by the NIH, the National Institute of Allergy and Infectious Diseases, in 2004, with the goal of really providing a free and public platform to enable researchers to access the growing number of types of data and the growing quantity of data on genomics and other related data. And they're mainly decided to focus on category A through C infectious diseases and tailor bioinformatic tools to the discovery of and study of these diseases. And as part of our mandate, we do outreach, which involves bioinformatics training, both at BRC sites and scientific conferences, or if your institution has a specific training request, we also tailor bioinformatics courses to that. And we also respond rapidly to new and emerging pandemic threats. So if you join us next month, you can hear what we've done in relation to coronavirus. So right now, there are two main BRCs, bioinformatics resource centers, that cover several different databases, and we are part of the Bacterial and Viral Bioinformatics Resource Center, which includes the Influenza Research Database, VIPER, the Virus Pathogen Resource, which we manage, as well as our sister site, Patrick, which focuses on bacterial pathogens. VUPathDB is another BRC that you'll be hearing from in this series, and FungiDB will be talking to you about fungal respiratory infections, and they mainly focus on eukaryotic pathogens and vector and host informatics. So just as a quick refresher, if you're not already familiar with Orthomyxoviridae, they're under the Ribaviria realm. They are negative-stranded, single-stranded, and segmented RNA viruses belonging to the Orthomyxoviridae family. They're enveloped, rounded, but they can be filamentous, and they're usually between 80 to 120 nanometers. As I mentioned, they're segmented, and then depending on which virus you're looking at, they can range between 6 to 8 segments. The segments length are usually between 1 to 2 kb, and the total genome length ranges between 10 to 13.5 kb, so relatively small for a viral genome. There are 7 known genera in the Orthomyxoviridae family. The most famous ones, obviously, are the influenza viruses. And at IRD, we focus on influenza A, B, C, and relatively new, we've added influenza D viruses. The other three families are not in IRD, and these are not generally human viruses, but they can infect arthropods and other vertebrates. So just quickly going over the genome structure of these viruses, just to give you a quick idea of what's encoded in them. Influenza A virus and influenza B virus both have 8 segments, and they managed to pack a lot of genes into these segments through a couple of clever different methods, such as alternative splicing, ribosomal frame shifting, or leaky transcription scanning. Influenza C and D, on the other hand, have a smaller genome, only encoding 7 segments and fewer genes that we know of so far. So viral genes are expressed through viral RNA polymerase that's encoded inside the virion and comes prepackaged with it. They use a clever method called 5' cap snatching to prime transcription. So taking the 5' cap from human or host mRNAs to prime transcription of the mRNAs from each segment, and then it can polyadenylate them using stuttering of the poly-U track that's encoded in them. And as I mentioned, there are a couple of extra variant genes that are encoded in these genomes through either alternative splicing, such as M2 and M42 and NEP, which you can see over here, for example. And then influenza A virus contains a ribosomal frame shift, which allows it to encode the PAX protein. And then there's also a couple proteins that are produced by just proteolytic cleavage of a larger polyprotein, such as M1 and B2. Okay. So this image is just to give you an idea of the Virion structures and what they're quite similar in these different viral types. However, there are a few important differences. And one of the main differences is in the ion channels that they use for acidification of these vesicles. And the M2 ion channel is found in alpha influenza virus, whereas beta influenza virus encodes a BM2 and NB ion channel. And influenza C and D viruses both have a CM2 ion channel. Another important difference between influenza A and B viruses and influenza C and D viruses is that while influenza A and B both have the hemagglutinin and neuraminidase proteins on the outside of the virion that facilitate binding and binding of the virion to the host cell and later budding through cleavage of the sialic acid with neuraminidase, influenza C and D have one protein called hemagglutinin esterase fusion protein that performs both of these functions. So just something to keep in mind if you're not already familiar with these viruses. So as I mentioned, HA and NA are only found on influenza A and B, and HA is a class 1 fusion protein that facilitates attachment of the virion to the host cell through binding of several different types of sialic acid receptors, and which one depends on the virus and the host cell. And influenza A virus is known to have at least 18 different subtypes so far, and these can be roughly divided into 2 groups. Neuraminidase, on the other hand, functions to cleave sialic acid and enable budding of the cell, budding of the virion from the cell. And it's known to have 11 subtypes. So between the 18 and 11 subtypes of hemagglutinin and neuraminidase, you can have a total of 198 potential combinations, which explains a lot of the diversity that we see in this virus. So the replication cycle, just real quick for this virus, starts with adsorption of the virion to the host cell, to the sialic acid over here. This is then endogenized into the cell. Acidification of this vesicle leads to uncoating of the virion. Viral particle, genomic segments from the viral particle are then transcribed using the viral RNA polymerase. This then produces both new proteins and new segments, new genome segments. And once they reach critical mass, these are packaged and start to bud off the host cell. And finally, the neuraminidase comes in and cleaves off the virion, allowing virus release. So scientists have developed antiviral drugs for almost every stage of this viral life cycle, including M2 ion channel inhibitors such as adamantanes, neuraminidase inhibitors such as oseltamivir and zanamivir, which I'm sure most of you have already heard of, polymerase inhibitors such as favipiravir, and the newest one that's been approved, which is a PB2 capping inhibitor, baloxavir. And if you'd like to learn more about these drugs, you can use IRD's antiviral drug search function to explore these some more. So like most viruses, once you develop antiviral drugs, they start to develop drug resistance. And again, we have a few different tools on IRD in which you can use to explore this. One of them is the phenotype characteristic search tool, in which you can search for viruses that have resistance phenotypes, or you can submit your own sequences for antiviral risk assessment. So influenza has a wide range of hosts, and the type of host that it infects really depends on which subtype it is. So influenza A virus is the one that's known to infect the widest variety of hosts, and this includes many non-human mammals, including marine mammals, many avian species, and of course a lot of domestic and agricultural animals as well. Influenza B virus is known as mainly a human virus, but it can infect some marine mammals. Influenza C virus can infect humans and pigs. And influenza D virus so far has only been detected in non-human mammals and has not been detected in humans yet. So through IRD, you can explore, if you're into more molecular epidemiology studies, you can explore the surveillance data that we have available for both humans through our human surveillance search tool, or if you'd like to look at animal surveillance in avian species or non-human mammals, you can look at our animal surveillance feature, and we have a nice mapping tool where you can map these different results on. Just a couple of different types of influenza that I'll mention because they just are well known and therefore have specific tools that have been tailored towards their study. Avian influenza virus, which if you haven't heard of, generally refers to influenza A virus, and it can be classified as either high or low pathogenicity. So for example, HPAI, highly pathogenic avian influenza viruses are, for example, H5N1, and IRD provides a tool for classifying H5N1 clades for that as well. And we also have a couple of tools for swine influenza, which typically includes strains of influenza C and influenza A viruses, such as the H1N1, and we do have a swine H1 clade classification tool. So moving on to the evolutionary, what's known about the evolution of these viruses, they engage in a couple of different mechanisms to kind of, as they constantly evolve. And one of them is known as antigenic drift. And this is kind of characterized by a gradual and continuous changes such as point mutations, and it's generally in the same subtype, and it may result in epidemics of influenza virus. And all influenza viruses A through D can engage in this phenomenon. Antigenic shift, on the other hand, is exclusively in influenza A viruses, and this refers to genetic reassortment of gene segments or transmission from birds or other mammals to humans. And it's characterized by major and sudden changes, and it can result in explosive spread. And again, we have a few different tools which you can use to study the evolution of influenza virus on our page, which I will go through later. So I'm going to end on this summary slide of the viral characteristics of the different influenza viruses, and see if we have any questions before we shift to our guest lecture. So we have a question, why do only influenza A viruses undergo antigenic shift?
So because there are multiple subtypes of influenza A viruses, many different hemagglutinins, and you don't see that with influenza B, it is possible for antigenic shift to occur. And an antigenic shift in that case would imply that a new HA for which the population is naive enters into that population. For example, in every pandemic of influenza that we have had, essentially we have had the introduction of a novel HA subtype into the human population. Now there are some exceptions, and or at least one exception that we know of, which is the pandemic of 2009. So until 2009, the concept was that for a new pandemic to occur, you would need a new HA subtype to enter into the human population. But in 2009, although we did have H1N1 subtype circulating in humans, a new H1N1 antigenically very different from the one that was circulating managed to enter into the human population. So although that wasn't strictly an antigenic shift, that was essentially the introduction of an antigenically distinct HA subtype into the human population. And if you take this into any in pigs, for example, you can see examples of introduction of novel subtypes into the pig population, usually from humans to pigs. But sorry, I made my explanation too long, but I hope.
Okay, so if we don't have any, there is one other question. Could the subtypes recombine under certain conditions?
So the issue of recombination with influenza viruses is sometimes a little bit controversial. There are papers from groups in Australia that suggested that maybe the 1918 H1N1 virus emerged through recombination of the HA. I'm not sure that the information is, I'm not sure that we can agree with that. Now, in other subtypes, the best example of recombination, and perhaps the only example in which recombination can occur, is with the H7 subtype in poultry. So both H5 and H7s can become highly pathogenic influenza viruses. And the mechanism of that is usually involved RNA structures and an stuttering of the polymerase during replication of the HA segment. But only in the HA subtypes there are examples of recombinations with either a ribosomal RNA or with recombination with other flu segments, the matrix and MP specifically. Those are the ones that we know of. And those usually result, the end result of that recombination event is essentially the same, is the accumulation of a polybasic amino acid region that allows the virus to cause systemic infection in chickens. Those are the H7 is the only example that I know of in which recombination, you can call it recombination per se.
So I'm going to move on to just a quick live demo of the site, the Influenza Research Database. And I'd like to invite you to either send us some feedback, or you can put your messages in the chat if there's any particular search tool or analysis tool that you'd like more information on. And I will work on making a mini tutorial and post it on YouTube, because it's virtually impossible to cover everything that we have available on the IRD site in half an hour or 20 minutes. So just to quickly tell you a little bit about us, this website is completely dedicated to influenza virus research. We aggregate data from multiple different sources. If you'd like to find out more about those, you can click under Sources of IRD Data in the About Us tab. But basically, we collect data from public databases such as NCBI, UniProt, or the Protein Data Bank. But we also add a lot of value through our own pipelines, and we generate our own data, such as through predicting CTL epitopes, or sequence similarities, or annotating proteins, or a lot of metadata curation and sequence curation in order to really tailor this database to make it easy for users. And of course, we also have data that's submitted to IRD directly. Because we don't have that much time, I'm going to just show you the three main sections of IRD. You can basically search for data, you can explore our different analytical tools, and we also have a workbench where you can save the data sets that you look for, or analyze your data through them, or you can share them with collaborators. And finally, you can also upload your own data, integrate them with the data that's on the website. And we even offer ways for you to upload the metadata so that you can integrate that into your analysis with the metadata that's already available on the site. So just clicking on the different IRD search tools, I'm going to just list these and give you a quick example of our most commonly used feature, which is searching for sequences and strains. And for that, we you can see in this red number how many sequences we currently have on the site. So right now it's just over 700,000. And you probably want to drill down to the specific sequences that you're looking for. And you can do that either by selecting a gene segment if you're only interested, for example, in HA, or you can select all segments if you want to complete genomes. You can click on this option if you're only interested in complete genomes, because a lot of people might only sequence the HA or the NA of a virus to just characterize it. So if you're interested in only complete genomes, you can click on that, and that reduces the amount of viruses that the database will retrieve for you. If you're only interested, let's say, in an HA segment, you can also click on this option here, which again will only select complete HA segments for you. We have options where you can select the type of host that you're interested in, especially if you're interested in non-human hosts. And again, this is where the value of IRD comes in, because we curate all of the metadata so that you don't have to sift through the scientific names and the common names. They're all just standardized to the common names, which personally I find a lot easier to remember. And we also have larger groupings. So let's say, for example, you're only interested in avian influenza. You can just go ahead and click on avian and then drill down through there into the specific species that you would like, or into the larger phylogenetic grouping of the avian species. So if you're only interested in ducks, you can click on Anatidae. If you're only interested in shorebirds, for example, you can click on shorebirds, or if there's a single species that you're interested, you can click on that. We also provide searches through geographic groupings by continent and by individual countries. And you can also search by virus type. if you're, let's say, only interested in influenza A viruses, B or C, and we've recently also added influenza D. If you have a specific subtype in mind that you're interested in, you can type in your subtype here and search for that directly. And I forgot to mention that if you're interested, let's say, for example, in the United States, you can also search by specific states. So moving on, you can, as you can see, 700,000 sequences is quite a lot to go through. So you really might want to shrink your data set to something a little bit more specific. So you can search by strain name. And then another thing that's unique about IRD is that we pre-compute clade classification. So for example, you can choose by global clade H1, and this will give you a drop-down menu if you already know which subclade you're interested in for the H1 viruses. Or if you're specifically interested in H5 clades, for example, you can also choose a specific clade that you'd like. You can again specify your date range, and I would encourage you to click through the advanced options and explore them, because there's a lot of different really useful options that you can choose from over here. And one of the things that you might be interested in doing is excluding lab strains, especially if you're doing molecular epidemiological studies and you don't want these kind of artificially made lab strains cluttering up your data sets. You can also remove duplicate sequences, which again, if you're only interested in looking at the diversity of sequences in a certain country, can really help reduce your data set. You can specify the minimum segment length if you're performing a phylogenetic analysis and you require a specific length of a certain segment. You can search by keywords, and if you're only interested in a specific flu season, you can click on the flu season that you'd like over here so that you don't have to customize the date range yourself. If you have more specific time in mind, you can choose the month. And you can customize the display fields that you'd like to see. And a lot of the, or at least some of the data that we have in our database also has some host information associated with it as well, such as specimen type, the gender, or the health status of the host, or often how the detection method that was used. So I'm going to just go back to the search tools and just give you a quick overview of what else is there. And again, invite you to just leave some comments in the chat if you'd like me to make a quick tutorial on any of these tools. We have some animal surveillance data that you can click through. And this is quite extensive, actually. There's over 1 million records for both avian and non-human mammalian species. So you can, again, I encourage you to click through that, and don't forget to look through all the different advanced options, which again can really help you tailor your data set into something that's most useful for you. If there are any structural biologists out there, you can look through our 3D protein structural files. If you have a specific mutation that you're interested in and you want to see how it affects the structure of your protein, you can search for the influenza A structures here. Let's say, for example, HA, and this will give you a table of results. You can find the relevant structure, and using our integrated structural viewer, you can kind of find the point that you want in the protein. And I'm not sure why it's hanging right now, but you can compare it to, you can also look at conserved proteins in the structure, and you can rotate this, and it's quite dynamic, and it's really useful for giving you an idea of where your region of interest might be in and how it will, how a certain mutation might affect your protein function. So again, there's so many different options here, it would be impossible for me to go through. But if you want a tutorial, you can just leave me a little note in the chat. Going back to the search tables, we have, as I mentioned, also some human surveillance data. We have extensive host factor data if you're interested in looking at host factor response. And in fact, the viral family that we have the most host factor data for is the Orthomyxoviridae. We have 51 data sets, and these are heavily and extensively and extremely carefully curated data sets. So they really have been vetted so that, and the data has been standardized so that you can easily go through these and explore the results of these different host factor experiments. Again, if you're interested in exploring the different types of antiviral drugs that are available, you should click through this option. If you're interested in the immunological side of influenza virus infections, you can explore our immune epitope database. And these have been sorted by B cell, T cell, and MHC class binding epitopes. They can be either experimentally determined epitopes or just predicted in silico through using the NetCTL program. You can search by subtype, by virus that you're interested in, look through whatever specific segment that you want, and again, search by host. And of course, there's a couple extra goodies in the advanced options. One of the things that I also like in this search functions is that you can search by phenotype. So if you're interested in looking at in retrieving all sequences that have a certain type of resistance and looking for mutations in those sequences, you can do that by just clicking on, let's say, adamantane or oseltamivir resistance. If you're interested in sequences that have increased virulence or increased transmissibility, you can also select those options there. And of course, again, you have the option of selecting by virus type, species, and location. Okay, so, and then one of the last things I'm going to go through on the search page is something that Catherine Mackin was at the University of Auckland, and other authors have worked extensively to kind of curate is the sequence feature variant types, which basically have characterized any regions of importance in these viral proteins. And if you'd like to find out more about them, you can just click on this publication that's linked over here and just read through it to kind of understand the different sequence features that were pulled out. And basically, they can be broken down into a few different feature types. Those that are, for example, immune epitopes. And again, you can sort through which type of targets or MHC class, whether they're functional epitopes that have to do with, for example, antiviral resistance or transmissibility, sequence alteration, or structural features, for example, like an alpha helix or a beta sheet. And we use these throughout the site for a lot of our different tools. So it's quite interesting. And if you'd like to kind of go through the list of different sequence features, you can find out more about them in the documentation that's linked through either this page or during our sequence feature, identify sequence features in segments in the search tools. So there's not that much time left, so I'm just going to highlight a few of our analysis tools on our Analyze and Visualize page. And as I mentioned, we have some tools that are specialized towards certain types of influenza, but we also have traditional or more commonly used bioinformatic tools such as BLAST, PCR primer design, sequence format conversion. We have some comparative genomics tools that will allow you to, for example, align sequences into multiple sequence alignments, view them, generate phylogenetic trees. If you're not an expert in phylogenetics, don't be intimidated. We have model compare tool that will help you kind of select the best evolutionary model for your data. Our tree viewer is very intuitive. It's with just a single click of a button, you can kind of colorize the branches on your tree by the metadata that's found in Viper. So let's say you just want to colorize it by host, country, or time. And then we also have a couple of tools that will help you analyze and look for sequence variation in your data set. For example, this traditional SNP analysis tool, or the Metacats comparative analysis tool for sequences, which I really like for influenza because it can help you detect issues such as antigenic drift. For example, if you have two different data sets of influenza from one year versus another, and you can just compare them and statistically compute whether there's any significant differences between last year's sequences and this year's sequences. Or you can look for evolutionary changes that might affect host infectivity. For example, compare a group of sequences from chickens versus a group of sequences from humans. So that's quite nice, and I encourage you to explore it as well. We do have some clade classification tools. As I mentioned in the introduction, our highly pathogenic avian influenza virus H5N1 classification tool. And this is based on the WHO's classification scheme, which you can find out more about by clicking over here or reading our SOP documentation. But basically, you can just either upload your sequences or just paste them in and click on run, and it'll assign your sequences to a specific clade. And the same is true for our swine H1 clade classification tool. And this is based on the swine H1 clade classification system that was published by Anderson et al. From Amy Vincent's lab at the USDA, who spends a lot of time studying swine influenza. So it's quite similar. You can either just upload your own sequences or use a data set from the database. So let's see, what else would I like to highlight in the remaining time? So we also have an HA subtype numbering conversion system, which is useful if you're trying to compare HA sequences, for example, from one subtype, let's say H1, to another subtype, H15. And this is based on the publication by David Burke and Derek Smith. And what it basically does is helps you relate amino acids that are structurally and functionally equivalent across all different HA subtypes using a numbering system based on mature HA sequences. So if we take a quick look at this figure, for example, let's say you're interested in this cysteine or at position 90 in H1 viruses. This tool will help you convert it and figure out that it's position 96 in H14 or position 87 in H15. So you can read through this paper if you want a little bit more detail on that, but again, it's quite easy to use. You can just either use your own file or paste in your sequences, and you just select which HA numbering scheme you'd like to convert it to, and then just press convert residue numbering. We have our antiviral resistance risk assessment tool, which I believe I mentioned again in the intro. And again, you can just input your sequences and we will calculate the antiviral resistance risk and give you the results back. And then one last thing I want to highlight before I leave some time for questions is, again, our... Sure.
I would definitely highlight the workbench as well.
Oh, okay. The sequence features, which are really important, but have a lot of great documentation. And the last thing I'm going to do is highlight the workbench, where you can save all of your experiments. What's nice about it is that they're really well integrated, so that you can click on an individual results, for example, and you can directly use the analysis tools on them to, for example, if you have a set of genomes, you can use a multiple sequence alignment tool, or just with one click on them and create a phylogenetic tree. You can also upload your own data, integrate them with those that are in Viper. And you can even upload your own metadata, which is really useful if you're trying to track molecular epidemiology. So I think I'll stop there, unless there's something specific that you think is important for me to highlight, and open it up for questions. Okay, so I'm going to just remind everyone before you rush off that we also have several other meetings coming up in the next few months. And these are all focused on respiratory pathogens, and they span multiple different BRCs. So let's say, for example, if you're working in a public health lab and you have a broad interest, we have lectures coming up on fungi, on coccidioides and aspergillus. We have more virus lectures on coronavirus, RSV, and enterovirus coming up. And our partners at Patrick will be discussing antimicrobial resistance in bacteria, and as well as bacterial respiratory pathogens. So you can find out more about these by just going to our homepage and clicking on Respiratory Pathogen webinar series and signing up there. If there's something in particular on the website that you want more information on, please feel free to send us an email by just clicking on support and ask a question, and we're usually pretty responsive and try to get back to you as soon as possible. And again, I would love to make a customized tutorial for anyone who has a specific tool or search function that they're interested in.